Okay, this is part 2 of lecture 5 on tumour suppressor genes and oncogenes, and what I'm going to cover now is the mechanisms of oncogene or proto-oncogene activation. So a proto-oncogene is just a normal cellular gene that typically controls cell growth in a positive manner. And when they become constitutively active, these genes become oncogenes because they are cancer causing or cancer driving. And the sorts of things that can happen with oncogene mutations is we get gene amplification. So you get additional copies of an oncogene, and you will have come across this, and I will cover this in a later lecture. Particularly the epidermal growth factor receptor and HER2 genes become amplified. And I'll talk about this throughout this lecture. We can also look at activating point mutations, typically by Ras. You can also have some of these oncogenes increased by viruses, so the human papilloma E5 protein will increase epidermal growth factor receptor. That would drive proliferation. We can get activation of these genes by translocation, so this is where chromosomes break, recombine, and you end up with a highly active promoter next to an oncogene. And then we can have fusion genes, which I'll explain at the end. So firstly we'll look at oncogenes as targets for chromosomal amplifications. And I mentioned in the previous module, probably pathological basis of disease, that tumours often duplicate genes, and we want to sometimes measure that to assign therapies. And I'm going to talk about this in more detail in the stratified cancer medicine session at the end of the module. But to cut a long story very short, the HER2 gene often becomes duplicated in breast cancers, and the EGFR gene becomes duplicated in many cancers, and basically you get multiple copies of those genes, which drives high level protein expression, which drives high level growth. And as I've explained in previous modules, this is a fluorescence in situ hybridization experiment, and in red is a DNA probe detecting the HER2 gene, and in green is a control chromosomal region. You can see there's lots of copies of the HER2 gene, so this is a HER2 positive breast cancer. And we'll talk about why these are treated with Herceptin later in the module. But the sorts of gene amplification that you see here leads to protein overexpression here. Now this slide might seem quite complicated, but we've covered some of it already. And it shows normal growth factor signaling, and also shows where oncogenes and tumour suppressor genes become mutated to affect normal growth factor signaling. So up at the top we've got a mitogen that binds to a receptor. That could be epidermal growth factor binding to epidermal growth factor receptor. That signal is passed on to a signalling cascade which includes Ras, and the MAP kinase cascade also includes RAF in there. We're going to talk about RAF in a few weeks' time. The end point of this is switching on of a transcription factor called MYC. MYC is an oncogene. MYC drives cyclin D. Cyclin D is an oncogene. Then cyclin D can help to drive cyclin E. Cyclin E is also an oncogene. The key point of cyclin E expression is that it phosphorylates PRB. PRB is a tumour suppressor gene, releasing E2F transcription factors. E2Fs are oncogenes. So what we can see is everything in green here is driving proliferation, everything in red is trying to stop it. So within any individual cancer there will be a mutation or overexpression of either the mitogen receptor, or Ras, or RAF, or the other MAP kinases, or possibly overexpression of MYC, possibly overexpression of this, cyclin D, possibly overexpression of E2Fs, or possibly loss of PRB, or possibly something else that does a similar job. This pathway will be defective in every single cancer, but it will differ by different tumour types. For example, lung cancers tend to overexpress EGFR, breast cancers tend to overexpress HER2. As we're going to see in a moment, thyroid cancers tend to mutate Ras. Melanomas tend to mutate RAF, which is about here. Lots of hematological cancers overexpress MYC. So something on this pathway is defective in every cancer. It just differs between individual cancer types. So on the previous couple of slides I've mentioned about EGFR overexpression, as shown here. The second mechanism of oncogene mutation is point mutations. Now these are always missense mutations, and point mutations change one amino acid to another. So here glycine to valine is at codon, specifically at codon 12 or codon 13. That specifically activates Ras. Now the Ras oncogene, which is here, it is in its signalling intermediate. It picks up a signal from the mitogen receptor and passes it on. If Ras is permanently switched on, then the rest of the pathway is switched on. So this is why Ras is so frequently mutated in cancer. And if codon 12 or 13 have been changed from a glycine to something else, that means that the protein is permanently switched on and permanently signalling proliferation. So Ras is mutated in about 20% of all cancers and 90% of all pancreatic cancers and the vast majority of thyroid cancers. So the reason that these mutations are always missense is because with proto-oncogene activation we need an active gene product, an active protein, and you couldn't have a nonsense mutation. That would be inactivating. So we're changing one amino acid to another amino acid. When it's a glycine, it's tightly regulated. When it's a valine, it's unregulated in terms of its ability to signal proliferation. So the third mechanism of oncogene activation is viral activation, and as I've sort of mentioned earlier on, the human papillomavirus has a gene called the E5 gene, and this helps to induce epidermal growth factor receptor accumulation on the plasma membrane. So on this little diagram here of membrane recycling, at point 1 I've drawn on some epidermal growth factor receptor on the membrane. At point 2 these get recycled back into the cell, and when they're no longer needed get sent for destruction at a cellular compartment called the lysosome. So EGFR receptors that are no longer needed get internalized in a vesicle and then get sent down here for degradation. However, if a cell is infected with the human papillomavirus E5 protein inactivates some of the crucial acid pumps that are in these lysosomes and these endosomes, and that means that these receptors get shuttled back to the plasma membrane. So in the cell that is infected with human papillomavirus, the E5 protein from the virus is preventing EGFR from being destroyed by the lysosome, and instead it ends up back on the cell surface. So you end up with EGFR accumulation, but so you get EGFR accumulation on the plasma membrane instead of EGFR degradation. So it's worth having a quick think. What do you think the mutation status is of the EGFR gene in cancers which are HPV mutated? Now in many cancers you get you get EGFR mutations which activate the receptor, or you get overexpression by gene amplification. What do you think happens when you've got human papillomavirus infection? So just have a little think about that. And this is the same question but just elaborated a little bit more. Given that HPV E5 causes EGFR overexpression, would you expect the EGFR gene to be amplified in cervical cancer as you see in lung and colon cancer? Would you expect it to contain EGFR activating mutations? Would you expect it to contain RAS and RAF mutations, which are downstream of the EGFR, which is where other cancers tend to pick up mutations? So just have a little think about that. Think about how tumours accumulate mutations, why they accumulate mutations, and whether you think these would be wild type or mutant for EGFR. Okay, moving on to the fourth mechanism of oncogene activation, and this is translocation. So I've mentioned the MYC oncogene earlier, and the MYC oncogene is here. So here's the MYC oncogene. The MYC oncogene is activated by growth factor signaling. The MYC oncogene, or the proto-oncogene, gets switched on, and that drives cyclin D. So that's its normal function, is to drive cyclin D. In many hematological cancers, and in this particular example Burkitt's lymphoma, we get recurrent translocations between two different chromosomes. So the MYC oncogene is on chromosome 8, 8q24, and the immunoglobulin heavy chain gene is on chromosome 14. And in almost all Burkitt's lymphomas, we get a translocation between chromosome 8 and 14, which brings together the promoter of the immunoglobulin heavy chain gene with the MYC oncogene coding sequence. So the promoter, which is highly expressed in these cells, in lymphocytes, particularly going down the B lineage, this gene is highly expressed. So this gene is highly expressed, so this promoter is highly switched on, which drives high-level MYC protein expression. MYC then drives cyclin D, and that drives these cells to proliferate abnormally, and that's what drives the cancer formation. And immunoglobulin heavy chain translocations are a common feature of hematological tumours of the B cell lineage. MYC translocations are also common in other leukemias, but often translocated to a different promoter. So wherever the MYC oncogene is translocated, it's translocated onto a promoter in an immune cell that's highly active in that cell. So it can be a different promoter, but it's often the same oncogene. Now this sort of phenomenon also happens in prostate cancer. You get a highly prostate-specific gene called TMPRSS2, or TMPRSS2 is its shorthand name, and that gets translocated next to the ERG oncogene, so you get androgen-driven ERG expression, and that drives prostate cancer growth. Now related to that translocation is translocations that involve a fusion protein. So previously you got a promoter driving a full-length coding sequence. Here we've got two coding sequences that are fused together. And this is chronic myeloid leukemia in the Philadelphia chromosome, translocation between chromosome 9 and 22, and I've covered this in pathological basis of disease, where I've covered FISH, fluorescence in situ hybridization. And what we effectively get is the coding sequence of the BCR protein forming the N-terminus of a large protein, and then the C-terminus comes from the ABL oncogene. So you end up with these large proteins made up of part BCR protein, part ABL oncogene, and this means that this ABL oncogene is permanently switched on, and that drives proliferation in chronic myeloid leukemia. So fusion transcripts from translocations are just sort of a variety of translocation-induced oncogene activation. So we've covered all of the main mechanisms of oncogene activation, and these occur in cancer, and we'll come back to some of these in the Stratified Cancer Medicine lecture, because we'll be looking at the effects of a H-Ras mutation in an EGFR amplified cancer. What I want you to have a little think about is the role of oncogenes in hereditary cancer syndromes. I told you earlier that tumor suppressor genes cause hereditary cancer syndromes. Why don't oncogenes cause hereditary cancer syndromes? There is only one that I know of, and that's multiple endocrine neoplasia. This causes autosomal dominant inheritance of thyroid carcinoma. So what I want you to have a little think about, and I can explain this more in the live session, is why would you not get hereditary cancers caused by mutated Ras or overexpressed EGFR in the way that you get hereditary cancers caused by loss of tumor suppressor genes? So just have a look at this. It's sort of this little sort of mental problem, but consider what would the effects of a Ras mutation be on embryogenesis compared to loss of one copy of a tumor suppressor gene, and consider it from the point of view of a developing fetus. So here's a summary of the key points that I've covered in these two videos. Tumor suppressor genes, there's a few questions that I want you to have a little sit down and think about. Why are tumor suppressor genes themselves recessive in activity but inherited dominantly? Why are sporadic tumours affecting only one site but hereditary or bilateral? Have a little think about the dominantly acting mutations. Make sure you're fully aware of dominant negative activity, and why oncogenes tend not to cause hereditary cancer syndromes. Okay, I think that's probably enough for this lecture.